The war on cancer began over 40 years ago with the signing of the National Cancer Act in 1971. Advances in therapy have prolonged life and, for some cancers, effectively provided cure. However, the decrease in the overall death rate has only been modest. For each 1,000 Americans alive on New Year's Day 1950, an average of about 2 people would die of cancer before the arrival of the next year. This statistic was similar in 2008, an overall cancer death rate on average of roughly 2 out of every 1,000 Americans. The improvement in the per capita cancer death rate appears particularly modest when compared with the more dramatic decrease in deaths associated, for example, with heart diseases. To hasten progress in basic biology and therapy, the National Cancer Institute began funding the Physical Sciences Oncology Centers, or PSOCs, in 2009. This is a $150 million 5-year program to bring physicists, chemists, mathematicians, and engineers together with cancer biologists, oncologists, and clinicians to look at cancer in new ways. 12 collaborative centers have assembled themselves to address a broad range of questions, including those related to heterogeneity, meaning variation between patients, between tumors, and within tumors, including cell-to-cell heterogeneity, evolution and evolutionary dynamics, instrumentation and measurement, and the development of conceptual laws and principles for understanding cancer biology and treatment. This video tutorial highlights 2 recent papers by authors from the University of California, San Francisco, working within the Princeton PSOC. The title of this video abstract is Dynamic Heterogeneity for the Physical Oncologist. We will present highlights from the first paper entitled Conceptualizing a Tool to Optimize Therapy Based on Dynamic Heterogeneity. The second paper, entitled Generalized Principles of Stochasticity Can Be Used to Control Dynamic Heterogeneity, argues that the concepts from the first manuscript can be used to understand a broad range of biological systems, including populations of interacting cells in tissue microenvironments, as well as organisms. Most of this tutorial is pulled directly from the first paper and inherits the Creative Commons license under which both papers were released at the journal Physical Biology. You may download the papers free of charge. This is the thread that this video abstract highlights. The timings of chemical reactions inside cells seem to display randomness. This stochasticity leads individual cells to execute back-and-forth transitions between cellular phenotypes. Sometimes a population containing individual cells undergoing phenotypic interconversion can exhibit reduction in population number, or indefinite maintenance of population number, or expansion, depending on whether drug therapy is administered sufficiently often. The authors begin with a perspective that explains how the timings of biochemical reactions can seem to display randomness. This seeming appearance of stochasticity can occur even in systems that are fundamentally deterministic. We are inside a cell. In this simplified cartoon, a target sequence of DNA becomes periodically shielded and then accessible as histone spools periodically condense and decondense. A copy of RNA polymerase moves in periodic lapse, spending some time away from the DNA sequence and occasionally passing nearby. A ribosome also completes a periodic circuit within the same cell. The relative spatial arrangements and velocities of the intracellular molecules change in an ongoing way. Occasionally, the histone spools might decondense and present the DNA sequence in an open format at a time when the copy of RNA polymerase is close enough for messenger RNA transcription. And occasionally, a ribosome might be in just the right place to bind a molecule of messenger RNA before it degrades. This combination of coincidences leads to protein translation. In this cartoon, the time periods of the movements of the histones, of the copy of RNA polymerase, and of the ribosome are incommensurate. They do not match up with each other. This means that the waiting times between consecutive protein translation events do not simply equal the period of motion of the histones, or the period of motion of the RNA polymerase, or even the period of motion of the ribosome. Instead, the waiting times vary, sometimes longer, sometimes shorter, in a complicated fashion that could be mistaken for a randomly generated pattern, especially when the periodic movements of the underlying molecular mechanism are not observed in detail. Monitoring translation events long-term might eventually reveal periodicity in the dynamics of this molecular system. At short times, however, the varied durations of time between reaction events resemble disorder. Using a second example, the authors note that deterministic systems can feature unpredictability. Configure two cells with identical initial molecular conditions. A strand of messenger RNA, a ribosome, and surrounding proteins shove past each other and alter each other's movements, leading, in this case, to the translation of protein. In the second cell, we slightly nudge the initial position of the ribosome to the right, preventing part of the chain reaction of molecular encounters that characterized the dynamics of the cell above. Even though the initial movements are similar in the two cells, the small discrepancy between the two initial conditions rapidly builds up in a biomolecular version of billiards, so that the outcomes soon become qualitatively distinct, with protein production in the cell above and the absence of protein production in the cell below. A biological cell can seem to display randomness, meaning messy patterns of reaction times. This can happen when the cell contains many components moving deterministically, even periodically, but according to a collection of periods that don't match. A biological cell can also almost seem to display randomness in the sense that two qualitatively similar cells can very quickly have two dramatically different outcomes. This can happen when the cells contain many components whose chain reaction interactions amplify small differences in initial conditions, preventing long-term detailed prediction of the cell's future configurations. It almost seems, not quite, but almost as though two cells in the exact same condition could have different outcomes. On this slide, we have presented two ways that timings of biochemical reactions can seem to display randomness. In the manuscript, the authors also discuss the contribution of quantum mechanical effects and the importance of low molecular copy numbers. In the next slide, we will relate apparent randomness in chemical reaction times to fluctuations in cellular phenotypes. A loose way to conceptualize the dynamics of a cell with apparent randomness in reaction timings is to consider rattling and shuffling a cell and then evaluating whether the molecular contents fall into an arrangement consistent with the occurrence of a chemical reaction of interest in a short interval of time. Reshuffling can lead to a different outcome, for example, the absence of protein production, as the cell forgets its previous configurations like a shuffled deck of cards. The likelihoods of drawing specific cards do not easily betray the order in which the cards were previously stacked. For a more mathematical description of stochastic processes, please watch the video tutorial on the stochastic simulation algorithm. As the cell is repeatedly shaken and its contents reshuffled, the translation of protein occurs at irregular time intervals, sometimes bunched together, sometimes sparse. This is one of the reasons that the level of a protein in an individual cell can momentarily explore peaks and troughs. We could draw similar cartoons for each of a large collection of proteins inside a cell. We have simplified this illustration by considering only one. To simplify the discussion further, we also replace the graded spectrum of protein levels with a telegraph signal that jumps between one relatively high and one relatively low protein level. The authors remind the audience that high levels of some proteins have been reported to protect cells from drug kill. The manuscript includes a specific example. For the toy calculation on this slide, the authors associate relatively high protein level with relative drug resistance and relatively low protein level with relative drug sensitivity. A cell thus makes transitions with apparent randomness between two phenotypes. This back-and-forth pattern is called phenotypic interconversion. In a population of cells, transitions can occur in different cells at different times. Multiple phenotypic states can be simultaneously occupied. In the manuscript, the authors describe two phenomena that can arise at the population level when constituent cells individually undergo phenotypic interconversion. The authors provide experimental examples for these phenomena, which they call the so-called generation of heterogeneity and, quote, homeostatic heterogeneity. Another way to visualize the seemingly random outcomes obtained by reshuffling intracellular molecular components is to represent the dynamics of the cells over a short time interval delta t as a round of roulettes. These drug-sensitive cells share the same wheel. This cell launches the ball, which by chance lands in the sector that prescribes maintaining status quo. This cell launches the ball and by chance lands on the instruction to convert to the drug-resistant state. This next cell happens to land on the sector indicating death. The cell after that maintains its initial state, and the remaining cell replicates. The population of drug-resistant cells plays roulettes using an analogous wheel with sectors possibly differing in angular widths. The wheels are reused to describe the dynamics of the cells during subsequent time intervals, also of duration delta t. This is called using a Markov model, and these processes are schematically represented using a flowchart with rate coefficients for replication, mortality, and phenotypic conversion that can differ between the drug-sensitive and drug-resistant cells. Should we view phenotypic interconversion as a challenge or as an opportunity for clinical treatment? On the one hand, phenotypic interconversion generates drug-resistant cells from drug-sensitive cells, suggesting that phenotypic interconversion is a source of therapeutic failure. On the other hand, phenotypic interconversion generates drug-sensitive cells from drug-resistant cells, suggesting that phenotypic interconversion is an opportunity for therapeutic efficacy. Should we view phenotypic interconversion as a challenge or as an opportunity for clinical treatment? On the next slide, we will use a graphical device the authors call a metronomogram to understand that, in some cases, a target population undergoing phenotypic interconversion can exhibit both shrinkage and expansion. The outcome depends on ensuring that therapy is administered with sufficient frequency and time. For this exercise, we assume we have a drug concentration that kills the drug-sensitive cells immediately, but leaves the drug-resistant cells completely unscathed. We want to show that in some cases, the population, meaning both drug-sensitive and drug-resistant subpopulations, can be eliminated even though drug kill never directly kills the drug-resistant subpopulation. Time runs from top to bottom. Drug kill occurs in an instant at time t equals 0. The targeted population just a moment earlier is a heterogeneous mixture of drug-sensitive and drug-resistant cells. Only the drug-resistant subpopulation survives. The surviving cells then proliferate and phenotypically convert, re-establishing a heterogeneous mixture of drug-sensitive and drug-resistant cells that confronts the next event of drug kill at time t equals delta t. The drug-resistant subpopulation survives. Our goal is that the targeted population shrink with repeated treatment cycles until the cell population is so small that, by chance, no cell is drug-resistant and the final residue of the targeted population is eliminated. TCD signifies the total course duration of all of these cycles combined. The difference between the number of cells confronting the event of drug kill at time t equals delta t and the number of cells surviving just a moment after the same event is the number of cells lost to drug kill. The difference between the number of cells confronting the event of drug kill at time t equals delta t and the number of cells that survives a moment after the previous event of drug kill at time t equals 0 is the number of cells gained through population expansion during the absence of drug. We want the number of cells removed by drug kill to exceed the number of cells gained through population expansion. Normalize, meaning divide both quantities by a common factor, the number of cells confronting the event of drug kill at time t equals delta t. The differences have now become fractions, which we label Fs and Fp shorthand respectively, for the fraction that has been sensitized during a drug-free rest interval, and for the fraction by which the population has expanded. Expressing these differences as fractions means that the maximum value we mark on each axis of a coordinate plane is unity. The authors call this coordinate plane a metronomogram. A metronomogram allows us to map the kill fraction and population expansion fraction for different drug administration frequencies. Above the diagonal, Fs exceeds Fp, corresponding to shrinkage of the target population. In this example, 5 cells confront the event of drug kill at time t equals delta t and only 1 survives, corresponding to a kill fraction of 4 out of 5 or 80%. The 5 cells that confronted drug kill at time t equals delta t are the progeny of the 2 drug-resistant cells that survived the previous event of drug kill at time t equals 0. The population increased by 3 cells during this drug-free rest interval, corresponding to a population expansion fraction of 3 out of 5 or 60%. This dot resides above the Fs equals Fp diagonal, confirming that Fs is greater than Fp and that the targeted population is shrinking, as illustrated in the cartoon timeline. For similar experiments with shorter intervals of rest between applications of drug, we would expect less population expansion, which corresponds to positions to the left on the Metronomogram. Higher frequency drug administration also abbreviates the time available for the drug-resistant cells surviving one event of drug kill to generate a sensitized population fraction, so the sensitized fraction is also decreased, corresponding to positions closer to the bottom of the Metronomogram. If instead intervals of recovery between applications of drug are lengthened, population expansion increases, corresponding to positions to the right on the coordinate plane. Given more time to recover following an event of drug kill, the drug-resistant population can generate a larger sensitized fraction before the next application of drug, corresponding to higher positions in the coordinate plane. As the drug-sensitive population is replenished, drug-sensitive cells begin to back-convert to the drug-resistant state. The curve bends, it stabilizes, and approaches a steady-state population fraction. For a more detailed description, please refer to the discussion of homeostatic heterogeneity in the manuscript. Positions above the Fs equals Fp diagonal correspond to drug administration frequencies that can reduce the size of the targeted population over time. Positions below the diagonal are expected to allow the burden to expand, and positions precisely on the diagonal can maintain the targeted population if the Markov model and its rate coefficients remain valid. In this simplified example, expansion, indefinite maintenance, and shrinkage are all possible depending on the frequency of drug administration. If the entire curve is known, patients and clinicians can try drug administration frequencies corresponding to red dots to determine whether toxicity can be tolerated. What strategies can we apply if the curve is not known beforehand? What can we do if the curve moves during a long course of treatment? What can we do when the targeted population harbors genetic mutations? This video has focused on reversible transitions between states differing in responsiveness to drug, but genetic point mutations are potentially permanent. In the manuscripts, the authors discuss strategies that might be useful for applying the Metronomogram in these situations. The application of these strategies may someday be part of the role of what the authors have called a quantitative oncologist, working with physical scientists, biologists, clinicians, and patients, quote, to optimally drain a tumor to extinction. To learn more about stochastic biochemical reaction timings, phenotypic interconversion, and the Metronomogram, including biological and clinical background, current limitations, and future opportunities, please download the original manuscripts available for free from Physical Biology.